Hello, and thank you for joining us for this Capsugel Dosage Form Solutions webinar entitled Enabling Taste Masking with New Technologies. It is part of our ongoing webinar series focused on innovative technologies for enhancing bioavailability, modifying release, and addressing other key formulation challenges such as today's discussion of methods for effective taste masking utilizing multi-particulate formulations. This webinar, along with our entire webinar series, can be found in the Knowledge Center on our website at www.capsugel.com and is available for on-demand views at your convenience. My name is Brett Caldwell, and I am the Director of Formulation Science at Bend Research, part of Capsugel's Dosage Form Solutions business unit. I will be moderating and presenting part of the webinar content today, along with my colleague Dr. Huell Williams, Senior Scientist with the Dosage Form Solutions team. Over the next 45 minutes, we will present two multi-particulate-based technology options that can provide effective taste masking alone or achieve taste masking while meeting additional target product profile challenges such as targeted release and or bioavailability enhancement. We hope to leave about 10 minutes to answer any questions from the audience. If you have a question, please submit it at any time during the seminar using the Ask a Question tab on the left of the screen. We will answer some audience questions at the end of the webinar today or follow up directly if we don't get to your question today. We'll start by briefly discussing the parameters of the taste masking challenge to formulators, then describe how multi-particulate technologies, specifically with case studies on lipid multiparticulates and osmotic bursting multiparticulates, can achieve taste masking as part of a target product profile. Anyone that has tried to feed a child or animal anything they know tastes or smells bad can appreciate a need for taste masking. Couple that with the stress of a sick loved one refusing a medicine that they need to help with their problems, and the challenge to a formulator becomes very stark in the realm of taste masking. Unpleasant taste reduces the palatability of dosage forms and is directly linked to poor medicine compliance, especially in specialized patient populations such as pediatric, elderly, or animal patients. The risk of taste masking affecting compliance is increased when treating patients who cannot or do not swallow traditional dosage forms, a phenomenon commonly called dysphagia. All palatability attributes should therefore be considered when developing a formulation with taste masking or easy to swallow. Taste or aftertaste can both result in a good first dose, but decreased acceptance for that dosage form beyond that is probably ensured. Odor can be a major issue with animal health formulations, and texture is a commonly misunderstood but very important aspect of a formulation also. The size of materials in a sachet, the smoothness of those materials, and even the nature of the materials being waxy and ductile versus something more inorganic in nature can certainly matter to the acceptability of the formulation. All of these palatability factors must accompany many additional criteria for a successful product, such as ease of administration, accuracy of administration, specifically the dose delivered, an adequate shelf life as always, and established safety of special excipients. Understanding the fundamental approaches to taste and masking taste gives the formulator a best chance of developing a formulation that meets all of those palatability and regulatory criteria. The fundamental approaches to taste masking start with a very simple concept, perhaps, of just minimizing the drug concentration below a threshold of sensation that's achieved through dissolution inhibition in a formulator sense. Taste panels and clinical trials can commonly provide a benchmark concentration for a formulator to work against in vitro. A second fundamental approach to taste masking would be to alter the free drug activity or concentration through complexation or partitioning into a separate phase. Something like a solution that is dosed to a pediatric patient can have materials in it that would complex and therefore minimize the amount available to hit the receptors on the tongue. One can also alter the drug diffusion to the taste receptors through viscosity modifiers, And one can also alter the perception of taste essentially through a balance of competitive binding. Those fundamental approaches are commonly attacked with common formulation approaches as listed below on this slide. Additives are very common, such as sweeteners, sugars, flavorings, and odorants to change the palatability of the material. Matrix entrapment is another common approach. Polymeric materials, gelling materials, or lipoidal materials can alter the free drug activity or the drug diffusion. Physical barriers and release modulation are also very commonly used. Physical barriers such as just a barrier coating on actives, on the active API alone, on the tablets or mini tablets for dysphagic patients. Multiparticulates can be easily coated, and merely including those materials in a capsule can commonly provide a heavy level of taste masking. Finally, release modulation is a very general approach that's used. The most general being usually a pH-dependent coating, but for appropriate drug chemistries, ion exchange resins, prodrug formulations, and salt formations can also be effective. Every product profile will have its own unique tolerance for these criteria on page 6, but here are some general key components for an optimized taste masked approach. Palatability, as we've described, we need good taste, odor, and mouthfeel for an acceptable formulation. Acceptability, all of these, again, a product will have its own acceptability criteria based on its market and intended use, but no cold storage is always desired, an ease of use. Fewer administrations is always more acceptable, and a long shelf life with convenient transport would be ideal. In the safety realm, we'd like to minimize the risk of local buccal irritation by the active and minimize any excipient levels to those absolutely necessary for a successful product. And finally, a platform that's compatible with a range of different drugs and finished dosage forms would be ideal. We've discussed all these earlier, but these might not yield an optimized efficacy on their own. If the audience takes away one memory from this webinar today, I hope it is that technologies yielding taste masking can also be enabling to efficacy also. For example, in the performance box, we can use multiparticulates to have immediate release or extended release profiles. They can have targeted delivery to specific regions of the small or large intestine, and they can use bioavailability enhancing formulations such as amorphous dispersions. So multiparticulate formulations are highly flexible in meeting any complex target product profile that's out there. And today we hope to tell you about lipid multiparticulates and osmotic bursting multiparticulate technologies with case studies that have been used to meet a range of these problem statements. Multiparticulate technologies cover a vast space of mechanisms of release, unit operations to produce them, and release profiles that can be generated from them. The mechanisms on this table are shown in the second column next to the cartoons, and they can include pH triggers, osmotic bursting time triggers, diffusion control, erosion control, or be immediate release in nature. The unit operations are shown in the next column, and these can be tailored to the equipment and available and the product need for any specific product envisioned. The profiles that can be generated are vast in nature and can utilize, as I noted, amorphous or crystallized forms and to generate taste mask formulations, immediate release, delayed bursts, or controlled release. Some of these can be mixed and matched by an experienced formulator with the right equipment for even more precise control over release if necessary. Today we're going to show some case studies on the lipid multiparticulates and the time-triggered by osmotic bursting multiparticulate mechanisms. With that, I'll introduce Dr. Huell Williams, Senior Scientist at Capsugel Dosage Form Solutions, who will talk through the lipid multiparticulate case studies.
Thank you, Brett, and hello to everyone on the call. So as mentioned, I will be speaking about lipid multiparticulate technology and its applications in drug delivery, and specifically where palatability and taste are key performance attributes. So to begin with, let me describe what lipid multiparticulates are. Well, in short, lipid multiparticulates, or LMP, are round, smooth matrix multiparticulates that are produced from lipidic excipients that are solid at room temperature. After production, the LMP are typically between 50 and 1200 microns in diameter, depending on the method of manufacture. Though it should be noted that when using Casagel's melt spray congealing process or producing LMP, we are afforded a much tighter control of the particle size distribution. One of the exciting aspects of LMP is that it combines some of the benefits of conventional capsule-based lipid formulations with the flexibility and additional functionality of the multiparticulate dosage format. Indeed, using our established non-aqueous melt spray congealing process to produce LMP, we're able to formulate the drug with a wide range of different lipidic excipients, including hard fats, waxes, and even surfactants. This flexibility in turn allows us to generate LMP that possess intrinsic taste masking functionality as a result of the hydrophobicity of the excipients that are commonly used. In addition, it is possible to generate taste masking formulations that offer sustained or immediate drug release profiles, or in other examples, taste masking formulations with solubility and dissolution enhancement. Our LMP formulation, therefore, offers significant flexibility and range, allowing a variety of different target product profiles to be met. This might be achieved through an LMP formulation comprising a single excipient or more complex multi-component systems, depending on the project and its objectives. In addition, LMP offers the opportunity to use high compound loadings up to 60% weight, while the smooth surface of LMP means that they are highly amenable to coating for additional functionality. Lastly, the fact that the formulation exists in a multiparticulate form means that this lipid technology is able to harness other benefits, such as good gut distribution, swallowability, as well as being compatible with a range of finished dosage forms, including capsules, sachets, and suspensions. In this slide, I've listed some of the common excipients used in LMP formulations. In short, excipients are all GRAS, which means that this technology can provide formulation options for a variety of market segments, including prescription, OTC, and H&N markets. And within these, LMP are also suitable for use in vulnerable patient groups, such as pediatrics. The core excipients of LMP include hard fats, though, as I've already mentioned, additional excipients may be added into the formulation to alter performance. For example, a surfactant may be added to the formulation to introduce self-emulsification properties, which is often desirable when the drug exhibits low aqueous solubility, while waxes or other hydrophobic excipients may be introduced to offer sustained drug release profiles. An additional point worth mentioning here is that our melt spray congealing method of LMP production allows us to produce lipid particles in the absence of binders such as lactose or microcrystalline cellulose. And this basically means that we can achieve high lipid concentrations in each particle and therefore have greater opportunity to use lipidic excipients to beneficially modulate performance. Moving on to some of the basic design principles of LMP formulations, the graphic in this slide describes fundamental LMP formulation properties when considering two inputs, the target release rate and the aqueous solubility of the compound. When taking this approach, the physical form of the compound and the intended mechanism of drug release are key properties to consider. For example, when aqueous solubility is high and a fast rate of drug release is desired, the preferred physical form of the compound is the crystalline form, as there is little advantage of solubilizing the compound within the LMP formulation. And in this case, a fast rate of release can be achieved by simple diffusion. If taste masking is needed here, the LMP may be readily coated for additional taste mask protection, as described in one of my later case studies. And this additional taste mask protection may be needed when the drug loading is high or where the product is mixed with food or a drink prior to administration. Alternatively, the LMP formulation may be tailored to achieve a more sustained release profile, where the rate of drug release is now governed by particle erosion, or alternatively by modified drug diffusion rates from the formulation, as I will describe in my second case study. Lastly, in instances where the compound exhibits low aqueous solubility, there are benefits of designing LMP formulations in which the compound exists in a solubilized state, while it's further advantageous for these formulations to contain a certain level of surfactant and digestible lipid excipient, so that the formulation can recruit endogenous bile salt and phospholipid mixed micelles for effective compound solubilization and absorption in the small intestine. So before moving on to the case studies, I hope that I've been successful in conveying the potential drug delivery applications of LMP in terms of providing sustained release, taste masking, targeted release, and solubility and dissolution rate enhancement. The two case studies I will now present are focused on examples where LMP formulations were used to develop palatable formulations for bitter tasting compounds. Though these case studies do capture other uses and advantages of this technology, namely the high drug loading capacity, the finished dosage form flexibility, safety in pediatrics, and Capsugel's established commercial pathway. The first case study relates to a small molecule compound that exhibited good aqueous solubility and was being explored by a company for the treatment of non-small cell lung cancers in children. Unfortunately, the compound was very bitter tasting, and due to compliance issues, simple and conventional liquid preparations could not be progressed in the clinic. A pediatric-centric formulation was therefore required, which offered better palatability and also immediate release properties in order to be bioequivalent to the existing solution preparation and also an existing adult capsule product that showed immediate release. In addition, the product needed to be developed at several different dosage strengths to enable accurate body surface area dosing, ideally via a direct mouth or sprinkle mode of administration. Based on all of these inputs, namely the need for taste masking, immediate release in pediatric subjects, and the bitter tasting compound, coated LMP was selected as the preferred technology approach to develop this product. The developed LMP product consisted of a lipid core and a hydrophilic pore former to provide immediate drug release properties following passage into the stomach and intestine. The LMP particle size was tightly controlled to 150 to 250 microns for optimal mouth feel, while smooth LMP were also aqueous polymer coated for additional taste mask protection in light of a high compound aqueous solubility, high loading, and the intended mode of administration. Aqueous polymer coating was preferred over solvent coating since the product was to be used in children, while the coating level was also minimized to ensure that acceptable excipient levels were not exceeded and to ensure that immediate drug release properties were achieved. With that in mind, two coating levels were progressed into a phase 1 study, and the lower 10% weight coat was shown to be effective in providing acceptable taste mask and overall palatability properties, while also showing bioequivalence to an existing immediate release formulation. And indeed, this LMP formulation is currently being used in phase 2 studies in pediatric patients. The second case study relates to the compound azithromycin, where there was need to develop a new single-dose product to be used in both adults and children to treat community-acquired pneumonia. Azithromycin exhibits high solubility in gastric fluid, which leads to rapid absorption from the small intestine. This in turn results in high peak plasma concentrations and associated side effects. So there's an opportunity here to develop a formulation that provided extended release to attenuate the rate of drug absorption for overall better tolerability. However, to complicate matters somewhat, azithromycin shows poor colonic absorption. So a specific release profile needed to be met so that bioavailability was not compromised by the slower rate of drug release and absorption. The full product was intended for use in adults and children, and an oral product, oral powder for suspension, was targeted as the final product concept. Azithromycin, however, exhibits a bad taste, so the formulation needed to show acceptable palatability alongside other product requirements, such as the extended release properties I've already mentioned, but also to be producible at low process temperatures to minimize compound degradation, and to only contain excipients that were suitable for use in pediatrics. Based on all of these inputs, LMP was selected as the preferred technology approach to develop this product. To meet the target release profile, the drug in its crystalline form was incorporated into a hard fat matrix that consisted of glycerol bahenate and a hydrophobic excipient for taste masking and for sustained release. And also poloxamer was also added to the matrix to fine-tune the release profile while also maintaining acceptable palatability. From this formulation, LMP will produce via the melt spray congealing process, which produced spherical particles with a medium particle size of approximately 200 microns, which is relatively consistent irrespective of whether the batch was produced at the small scale in the lab through to commercial scale batches. After the target release profile was met, systematic in vitro evaluation of the LMP was conducted to understand and delineate the mechanism of azithromycin release on hydration of the LMP particles. As demonstrated here, SEM analysis revealed that pores readily form within the LMP particle as it is hydrated. These pores stem from the dissolution of the drug in the poloxamer near the particle surface, resulting in void spaces within the impermeable lipid matrix. And in turn, this forced drug diffusion to occur through these large voids and interconnecting small channels. This slide speaks to some additional in vitro work that was carried out to systematically evaluate the effect of various LMP formulation variables on the azithromycin release profile. In the upper two panels, the results clearly show that drug release rate can be modified by changing the level of only one formulation component, either the level of dissolution enhancer in the LMP formulation itself, as shown in the upper left, or the level of basic excipient in the final suspension formulation, as shown on the upper right panel. The lower two panels speak to the capacity to modulate drug release rate by controlling particle size of LMP or the drug crystal size within the particle as well. In short, these dissolution results are critical to the overall understanding of the azithromycin LMP formulation, and were used to identify key formulation parameters that must be tightly controlled for consistent performance. In addition, these results also illustrate the capacity to effectively modulate the rate of drug release from LMP formulations without the use of external coatings. So to briefly summarize the azithromycin case study, LMP technology was used to produce a specific release profile for azithromycin so that there would be a clear reduction in peak plasma concentration of this drug following oral administration. This target was met as confirmed in the clinical results shown here in the upper right panel, where the LMP afforded a much more desirable pharmacokinetic profile when compared to an existing sachet formulation. In addition, following further in vivo testing, an in vitro in vivo relationship was established, as shown here in the bottom left, where in vitro release rate and overall in vivo exposure was correlated. And this overall confirmed the importance of meeting a specific release profile that ensured optimal tolerability in terms of side effects and optimal exposure needed for efficacy. Lastly, this work conducted at Penn Research in conjunction with Pfizer included significant engineering efforts and finally led to the product ZMax being commercially available in the US Japan, Canada, and New Zealand.
Thank you, Huel. I will start to talk about the multiparticulate approach by osmotic bursting, which is something that's precedented in the literature for time-triggered osmotic pressure release of drugs. It's engineered by a fluid bed layering process, typically. And fluid bed layered multiparticulates is a big, broad class enabled by the flexibility of fluid bed processing. It typically starts with an inert core of some variety that's commercially available. And on top of that, fairly anything that can be atomized through a nozzle can end up on a fluid bed layered core. That can be used to engineer an immediate release profile, as shown on the left side of this slide. That can be used for short Tmaxes or rapid onset. Can also use a pH-triggered release. This can be enteric or anti-enteric, reverse enteric in nature, to enable release such as taste masking to get through the mouth and into the stomach, or protect the drug from the stomach and acidic pHs until it gets into the duodenum where it can be absorbed, or it can be released later in the small intestine for better efficacy, or enabling once daily dosing if a second pulse is enabling in that regard. It can also be used for local therapies, releasing biologics and other materials later in the colon where they are desired for those types of efficacy. Extended release and extended delayed release are certainly capable to dampen a Cmax or reduce the side effects or reduce the dosing frequency of any particular medicine. And enhancing solubility and dissolution is still a viable option with the fluid bed layering. We commonly use amorphous dispersion layers to increase solubility and dissolution rate and or inhibit precipitation to enable extended release profiles. For this particular problem statement today, the problem statement was a fixed dose combination with 2 pulses being desired. These molecules both had good water solubility and were used for the treatment of psychostimulant abuse or the side effects therefrom. The performance needs, again, was a fixed dose combination of 2 drugs. For the ideal efficacy of these 2 drugs, we needed a separate release profiles. The first drug was immediate release in nature, and the second drug for ideal efficacy was released 3 hours post-dose. This could be achieved by a pH-triggered entity, pH-triggered coating, something that would release mid to late in the small intestine. However, that can be variable depending on the fed prandial state of the patient and interpatient variability of transit time. So with this particular use, we wanted to come up with something that would be more reproducible and less dependent on patient population changes. So the osmotic bursting for time-triggered release was seen as a definite advantage to the efficacy of this particular product. The multiparticulate format then was ideal to mix the 2 drugs at an appropriate dose to enable ideal efficacy and easily used in a capsule. The ease of that fixed dose combination mixture kind of worked for the multiparticulate and against any monolithic dosage form such as a tablet if all else was equal about the performance of the 2 products. Finally, appropriate stability was obviously required, and in this particular case the capsule provides any taste masking necessary, but engineering in the efficacy was the primary product need in this case study. So the cartoon and reality of osmotic bursting multiparticulates is pretty impressive to me as a formulator, that we can engineer something that looks like the cartoon and has the SEM micrograph of a cross-section as shown in the upper right. Starting with a fluid bed coated core, in this case the microcrystalline cellulose was used. This size was fairly big in this case because it was going to be involved in a capsule, but we've used much smaller cores for orally disintegrating tablet or sachet use, something that would require taste masking in the mouth. The next layer of the cartoon and the micrograph is a drug layer. This is where we made a slurry of crystals and applied it by fluid bed technology with HPMC as a binder. We had to tune the loading such that we would have suitable loading for the dose required and the capsule size of interest. And again, this can be an amorphous dispersion very easily if necessary for any drug application. The swelling layer is the next layer going from the inside out, and it's key to the mechanism of release. As water permeates the upper layers, this swelling layer starts to apply physical pressure on the barrier coating layer above it and effectively bursts it at a very prescribed time, as you'll see in the next couple of slides. The barrier coating layer is the final layer, and it does allow water to infuse into the particle, and this barrier coating thickness and strength is key to tuning the lag time of any formulation. Overall, the size of these particles does have some effect on the release parameters that need to be achieved for any particular product. So the osmotic bursting multiparticulates hallmark would be what's shown in the left side here. It's a very tunable lag that is only dependent primarily on the barrier coat weight, followed by a pulsatile to short controlled release duration. In this particular case, you can see the lag changing as a function of coating thickness from 15 to 18 to 20% coating. And that lag is followed by essentially 80% of the release of the drug within 2 hours after the lag time has been achieved. Sieving the size of these particles, sharpening that down or changing the size, does sharpen the profile or move it around as desired for any particular product. The mechanism of release is relatively insensitive also to a wide variety of parameters such as environmental pH. Showing the graph on the right side, what's shown here is at 2 different coat weights, low or high barrier coat weight, and at 2 different pHs, pH 6.0 versus 6.9, these particles can achieve the same release rate independent of pH. This is important back to my thought on the patient-to-patient variability, that if a patient population due to a disease state or their diet or their age have a different pH profile throughout their small or large intestine, these particles will still release at a very prescribed time. And that's, again, important to efficacy in this product and in other projects that we've had. The mechanism of release is also relatively insensitive to sweller layer thickness, surprisingly, even though that's the key to the mechanism of release. The swelling layer thickness is not critical usually. And even the drug properties are fairly insensitive. In this particular case, we could use 2 different salt forms of the drug, and we've also used amorphous dispersions underneath these of very highly lipophilic drugs, and seen very similar profiles to those shown on this page. A hallmark of the classic osmotic push-pull osmotic pump monolithic tablets is their strong in vitro and vivo relationships that come from the mechanism of release relying only on water activity outside of the tablets. When an osmotic bursting multiparticulate is well designed and formulated, these particles can also achieve a very strong in vitro and vivo relationship. Shown in this literature example is the in vitro and vivo relationship for in vitro release, the lag followed by the very pulsatile release, again, similar to the case study that I showed, just dependent on the barrier coating thickness, and then the in vivo concentrations that were achieved based on those different particles and their lag times that were generated by formulation. So in summary, multiparticulates can utilize such an array of mechanisms of release and materials that an ideal target product profile should be in reach. Capsugel Dosage Form Solutions prefers to work closely with client teams to connect any target product profile to a formulation design, then develop that together and ultimately manufacture those materials for clinical or commercial use or inclusion in other finished product ideas. Specific to multiparticulates, the early phases of a multiparticulate project can be led from capsugel dosage form sites in Bend, Oregon, or Strasbourg, France, then hopefully transition with success together to Greenwood, South Carolina for commercial LMP manufacturing. Capsugel also makes a wide array of capsules that can be used for multiparticulate dosing, including designs specific to sprinkle dosing for pediatric use or geriatric use or enteric protection of the materials inside. We also have in place specialized manufacturing, including FDA and MHRA accredited finished dosage sites that can handle highly potent, controlled substance, hormonal, and oncology compounds. High quality science and engineering is always core to these offerings at each stage of the product development cycle and has enabled the successful advancement of hundreds of compounds. Again, multiparticulates are one primary tool to achieve any sort of product profile, including modified release or special uses such as taste masking as discussed today. These can be coupled with almost any bioavailability enhancing paradigm or dosage form to suit any product need. Shown on this slide would be the bioavailability enhancement profile technologies that we commonly use and have man-years of experience with. The modified release and targeted release entities that we can use, and the specialized applications that we use those to target. In summary, the taste masking of bitter active ingredients and overall palatability considerations are critical for patient compliance of enhanced formulations. The specialized population compliance can be especially challenging where swallowability is a concern, and that can be true in the pediatric, geriatric, or animal health environments. Many formulation challenges today are not purely limited to taste masking, and hopefully the case studies today show that enabling technologies must be multifunctional and fit for purpose to achieve any product profile needs. Multiparticulates are uniquely suited to be flexible in meeting these types of problem statements. For example, combining taste masking with bioavailability enhancement or taste masking with a fixed dose combination that enables efficacy for a product. We talked today about lipid multiparticulates and osmotic bursting multiparticulates as highly flexible approaches to meet these formulation challenges. And capsugel dosage form solutions is available and to offer a full breadth of these technologies and capabilities to help achieve your product needs. With that, I'd like to open the floor to audience questions, and if at any time you'd like to view this webinar, please visit our Knowledge Center at www.capsugel.com. I see that we have several questions. If you have one, please submit it using the Ask a Question tab. Let me get started with the first question. What do you use for in vitro taste masking testing? We commonly use a saliva buffer that's out of the literature. There's a couple of recipes that are present out there. We commonly rely on threshold concentrations being set by the client or a taste panel or something like that has come from previous clinical trial experience, and try and minimize that threshold concentration we reach in any media. But we do tailor the test to minimize the amount of media, maybe thinking of 1 to 5 mils in the mouth. And we'll also, whenever we're using a bioavailability enhancement formulation, in the taste masking, such as a spray layered dispersion underneath the osmotic bursting layer. We can transfer that in ways and then understand that we've kept the high energy nature of the formulation. Another question that we have received is LMP technology applicable for both hydrophilic and hydrophobic compounds? Huel, would you like to address that?
Sure. So that's a very good question, and in short, the answer is yes. But if I can elaborate a little bit more. So for hydrophilic compounds, there may be a need for LMP technology for taste masking or, for example, sustained release properties. And within the LMP formulation, we are able to utilize the drug in a crystalline form, which might be certainly applicable for hydrophilic compounds. Now, for hydrophobic compounds, the technology is also applicable, but here there is a potential to utilize the drug in a dissolved state within the LMP formulation, so we can render the compound non-crystalline. Of course, this is dependent on the affinity of the compound for the lipidic excipient that we use, also the drug loading and the dose within the LMP formulation as well. With hydrophobic compounds, also we can work with crystalline drug, and in essence then you have a lipid suspension type formulation, but again harnessing that flexibility of the multi-particulate state.
Great. I've seen a couple of questions that are related to a similar topic. Are there any limits to the final dosage form that can be utilized with the lipid multiparticulate or osmotic bursting multiparticulate? I think the short answer to that, the generic answer, we would say is no. We've had a lot of success, obviously, rolling multiparticulates into any kind of capsule, whether it's an adult normal capsule or a pediatric sprinkle capsule. However, we've definitely used certain case studies, problem statements, target product profiles that came from clients where we have included both of these types of multiparticulates, including LMPs with coatings to increase the taste masking power, if you will, diminishing the threshold concentrations into formulations such as orally disintegrating tablets, or as Huel talked about earlier, into a one-time make at the time of use sachets.
So there's a question that's come in asking whether the melt spray congealed method of producing LMP can give rise to true immediate release performance, or is it dissolution retarded? So in short, the answer is that LMP formulations can indeed achieve very fast and immediate release properties. Although we're working with lipid matrices, the key thing to remember is here that there's a multi-particulate state, so we're afforded a high surface area for water ingress and drug dissolution and subsequent egress. Also, the LMP formulations don't need to be entirely lipid-based, as was the case with the Zmax case that I presented. There is a potential to include hydrophilic dissolution modifiers in the formulation to increase porosity and promote release through porosity, as I've mentioned, and also osmotic means by encouraging water ingress. So the LMP formulation can achieve immediate release properties with the possibility of this translating to immediate release in vivo.
Looks like there's another question on what is the smallest core to form a multiparticulate. This is somewhat, again, formulation dependent and target product profile targeted on a case-by-case basis, but we can commonly, for the LMP technology, we can usually make something in the effect of 100 to 400 microns fairly readily. When we're thinking about something that's taste masked, something that has good mouthfeel, we will definitely lean towards 250 microns or less. The LMP technology, again, because of its materials being soft and waxy, we get very smooth surfaces. You can usually, if you will, get away with a slightly bigger particle than you can with other types of granulations or matrix formulations, and end up with a good mouth feel for those particles. From the fluid bed coating side, we commonly use we've retrofitted a lot of our fluid bed coaters to start with cores that are in the 100 to 200 micron range and add layers as appropriate there.
So a couple of questions have come in with similar topics relating to the stability of lipid multiparticulate formulations. So if I could speak first of all regarding physical stability, this relates to both the compound as well as the formulation, since we're working with congealed lipidic excipients. So in terms of the compound, if the drug exists in a crystalline form, then we will typically monitor the physical form of the compound post-manufacture using X-ray diffraction to ascertain whether there's any polymorphic change or change in particle size on storage, although that can be attenuated through judicious selection of the process parameters. In terms of physical stability of the lipidic excipients, it's well known that there's a slight risk of lipidic excipients falling into a metastable state once rapidly congealed. And again, this can be evaluated using, for example, X-ray diffraction or DSC as well. And in terms of the LMP technology, we're cognizant of this as a slight risk, and we'll extensively monitor that during stability. Although it's worth pointing out that this can be addressed via a potential annealing step post-manufacture to basically render the LMP particle more physically stable, but also through judicious excipient selection as well as the process conditions as well. In terms of chemical stability of the LMP formulations, well, it's in many ways much like conventional lipid formulations, although it's worth bearing in mind here that we are harnessing the semi-solid solid state, so with reduced molecular mobility, one can expect enhanced chemical stability as a potential advantage of the liquid formulations, and also the fact that since we're able to work with the drug in the suspended crystalline form, we know, of course, that that is more likely to offer more favorable chemical stability, often compared to instances where the drug is in a solubilized state.
Let's see, there's a question here on does the MSC technology give rapid or true immediate release performance, or is it dissolution retarded? And the answer is it is totally up to the formulator and formulation client team. The somewhat, it is based on a diffusion commonly of water in and solution out, but through particle size, through drug solubility of any medium, and through the effects of coating, we can make them, we can tune the dissolution profiles of them fairly readily.
So a question has come in, which is, I believe, a follow-up to one of the answers that you gave in terms of generating immediate release properties in lipid multiparticulates, for example, with hydrophilic pore formers, and what impact does that have on the taste masking properties? Well, as you can imagine, if we're adding in excipient components to enhance dissolution rate, then in many ways that can be detrimental to taste masking, particularly if, for example, the mode of administration is to administer the formulation prior to a drink or a food prior to administration. Direct to mouth is probably a smaller window to cover the overall taste masking properties. So in instances where there's a need for immediate release properties and taste masking with LMP, this is one of the examples, as I alluded to in the slides, where an additional coat can add some robustness and functionality in terms of the taste masking capability of the LMP. So that's one of the challenges in terms of having immediate release with taste masking, and that's where the coating comes into play. The advantage of LMP compared to other coating techniques is the smooth surface of the LMP, basically making it highly amenable to coating. Also, it's worth bearing in mind that we have this capacity to have a high drug loading within the matrix particle itself, plus the GRAS excipients, and also the safety and biocompatibility, which is obviously desirable from a pediatric drug delivery point of view.
There's a question here on how to characterize and test the reproducibility of coating LMP formulations, and this is really no different than any other formulation in the sense that we use all the tools available to us. We commonly use a lot of microscopy. We commonly use a lot of dissolution testing and do a couple of different types of tests to make sure that it transfers so that we know that it's doing what we expect it to, and including things like we like to get to the fundamental parameters of the coating so that we understand what its limits are. For example, if you have a very low taste threshold but you have a porous coating material that might not be able to minimize the threshold as much as you'd like. So we commonly are tuning the surfactant, the plasticizers and things like that in our formulations to suit the formulation that we're making.
A question is coming regarding the melt spray congealing process to produce LMP. Basically, the key question here is what are the typical process temperatures that are used to produce lipid multiparticulates? So whilst this is somewhat formulation dependent, it's typically in the region of, let's say, 50 to 75 degrees Celsius, and reflecting the temperature at which we need to melt and liquefy the formulation. It's worth bearing in mind that this is a somewhat ambient temperature and moderate temperature, I should say, and we see that in the case data presented on the Zmax, where the compound in question did suffer a certain degree of thermal sensitivity. So in that regard, the LMP technology was highly suitable by basically minimizing the exposure of the compound to elevated temperatures for overall favorable stability profiles.
Looks like there's a question here on, are there any special requirements for the API particle size distribution for the LMP technology? I think the short answer is that if you're trying to make a certain particle property for your mouthfeel, there's definitely a limit that you'd have there. We definitely need them to be able to be coated fairly readily. However, we have seen fairly wide distributions form LMP particles fairly readily. One of the common, most fundamental parameter that we have to deal with is viscosity of the materials as they are put onto the spinning disc, for example, and that's not really too limiting a factor usually for loading or anything to that effect.
There's a short question concerning LMP again, and basically whether there are any commercial LMP products on the market. And in short, the answer is yes, and I can reference the Zmax product that was presented in my case study, which is azithromycin, which is a high single dose suspension available on prescription for both adults and pediatrics, where, as I was saying, the LMP formulation is primarily there to offer a specific release profile to attenuate side effects and also achieve optimal efficacy in terms of overall exposure. And within the suspension formulation, it's mixed with a certain level of sweetness to offer good palatability and taste masking. But the LMP itself offers some degree of intrinsic taste masking and palatability through the nature of the formulation being lipid-based and often offering this creamy mouth sensation as we would expect through semi-solid lipidic excipients.
There's a couple of questions here that are related to chewable tablets, I think related to animal health, but also obviously related to anything that's on the market for humans. The LMPs and the osmotic bursting multiparticulates can be used in chewable tablets. and we have some experience doing that, both for just a pure LMP or something that's been coated, for an osmotic or for an osmotic bursting multiparticulate that obviously has the coatings on it. And there's definitely a special choice of excipients that you have in that particular case. There's definitely some friability to consider for the tablet and the coatings, but just through a lot of careful studies, it's usually fairly amenable to that type of technology.
So there's a question regarding LMP technology and how it might differ from other matrix or encapsulation multiparticulate technologies. Well, the first point to get across and reiterate is that with LMP, we're harnessing lipidic excipients and truly lipidic particles. So alongside that, within LMP, we're able to harness the associated benefits of lipid-based excipients such as biocompatibility, safety, taste masking properties, sustained release, and potentially solubility enhancing. So we have a wide breadth, I should say, in terms of performance functionality with LMP technology, and alongside that, the associated variability in patient type in terms of adults and pediatrics and animals, as Brett was alluding to as well. Also, in terms of the process through which we produce LMP at Capsugel, that's the melt-spray congealing process, this is a non-aqueous way of producing lipid multiparticulates. And basically that offers us a greater flexibility in terms of the nature of the compounds, so we're able to work with very hydrophilic compounds, and also then high doses without concerns that this would limit encapsulation and yield as well during the manufacturing process. So LMP technology, the key thing here is that it harnesses lipidic excipients, so we can associate the advantages of lipid technologies with this particular platform. And then also in terms of its non-aqueous method, the production offers flexibility in terms of the components being both hydrophobic and hydrophilic.
Looking at the time, the last question we will field for today, it relates to the osmotic bursting multiparticulate technology. How does the liquid penetrate the barrier coating of ethyl cellulose to activate the sweller layer? The ethyl cellulose coatings are plasticized, but they are not, and they are integral coatings, but they do allow water to penetrate through them, and it's the physical force of the water coming in at certain rates, and it is tunable by other additives, so that is one handle of a formulator's control, as well as particle size to enable some control over the dissolution rates. But that water coming in very reproducibly and independent of pH usually swells the swelling agents and applies the physical pressure to burst any multiparticulate. Those are all the questions we do have time for today. If you have any follow-up questions, please feel free to contact us directly via email. We will be hosting additional webinars and hope you will continue to participate. In fact, we have two coming up. On February 10th, we are hosting a webinar titled Bioprocess Design and Operation, Enhanced Bioreactor Observability and Process Guidance. Then on March 19th, we will kick off our Fundamentals series covering our spray-dry dispersion technology. Please check capsugel.com for details and to register. On behalf of Capsugel Dosage Form Solutions, myself and Huel especially, I'd like to thank you for joining us today and for the excellent questions we received. Please fill out the survey that will pop up on your screens as soon as the webinar is over. Your feedback will be incorporated into the future webinars planned. Please contact us at the email address shown on the screen if you have any questions or comments, and a representative will follow up. Thanks again for your time, attendance, and interest today.